So the title originally was designing when the principles of operation are not clearly understood. But I took this from my friend Greg Worrell at the Mayo Clinic, and he likes to ask, when do we know when we know what we don't know? And I think that's something we have to think about in translation and be honest about. So once again, disclosures, starting from the left, these are all concepts that I'm going to talk about, drawing from some commercial product, but the purpose of this talk is to discuss research concepts, so of course that makes it investigational use only. And I want to thank some of our researchers who are using our tools, who have offered to share data from their investigational studies, and of course I'm an employee and shareholder at Medtronic. So let's start with just grounding the problem statement, if you will. This is a gentleman with essential tremor. It's an intention tremor. On the left, his device has been turned off. Sorry about that. And on the right, his device is turned on. So you can see, I can't get that to go in now. You can see that his tremor has been fairly well dealt with, but he still has some residual issues. So even though he's better at his activities of daily living, he clearly has an issue when his device is off. So let's go and get it there and click. Thanks. So what's inside of him? To kind of give you a feel for it, there's, the neuromodulation technology, at least from Medtronic, really did build on the cardiac rhythm management space. And so that's why our devices start out looking a lot like a cardiac pacemaker, but the leads, instead of being routed into the heart, were routed up into specific targets in the brain. Now, circuit designers, you got to ask yourself, how did you find those targets? Something that a lot of people don't think about. How do you know where to stick those electrodes? And that really came from bootstrapping off of classical lesioning targets from stereotactic neurosurgery. And so that was a good balance then of the technology and the existing clinical practice for stimulation-only systems. But really it left us with the conundrum that the mechanism of action is not clear. And that kind of sets us up for thinking about how do we improve a device? What are the opportunities in that space? And so I take a snapshot of 2015. This is from the Medtronic perspective. We have a series of commercialization devices and a series of systems that are still in research and development. And I highlighted two areas that I want you to think about. One, I'm going to focus on movement disorders for the rest of the time, just to be mindful of the clock. When we look at Parkinson's disease and tremor, I showed that example of a gentleman with a device. I think, what can we do to make that system better? So we don't have a clear mechanism of action. It's a little bit unknown. And what's interesting is, in a way, it's a victim of its success. And by that I mean a lot of clinicians say, open loop works. It reminds me of the first pacemaker. Many people said it was good enough. You didn't need sensing. That was actually said. It's taken for granted today that there's sensing in pacemakers. But there was a strong trend at that time saying it kept the patient alive, it did its job. And there's a little bit of that bias here. In fact, as engineers, part of me sharing my lessons learned is our expert panel actually chastised my team for thinking about closed loop and movement disorders. They said motion is not episodic. Epilepsy, that makes sense, but why would you ever want to close the loop in a movement disorder subject? So let's hold that thought. The other issue is when we think about new areas for exploration in neuromodulation, and specifically epilepsy. So you saw that gentleman with his tremor. So he comes into the clinic, and you can adjust parameters, and you get kind of an instantaneous feedback on how well your parameters have been set. Well, for epilepsy, it's more of an episodic event. So how do you know in the clinic when you're setting up parameters that you're actually doing the correct job? And so in the initial research, there was kind of a pull from parameters from vagal nerve stimulators for cycling parameters, movement disorders and basal ganglia circuits to apply to the thalamus, kind of a hodgepodge of parameter setting. And so there's this need to step back and say, from first principles, fundamentals, what is the most appropriate way to set up a device if you want to treat epilepsy? And what do we need to do to help the clinicians optimize that process? So I'm kind of a history buff. I like to think about history of technology. And part of it is we don't have to feel lonely. So we have hard problems in neurotechnology, but history is always full of hard problems. So this is about 100 years ago. this is when people are still learning how to fly. And I kind of like this example. You make observations, birds flap their wings, so if I'm going to build an airplane, clearly it needs to be flapping, doing its job. The other examples of, add more horsepower, all else fails, let's put more electrodes or turn up the amplitude. Even go back to Leonardo da Vinci and just take Leonardo's ideas and hook it up to a tractor. But then, now we start getting some hints. There's some promise that you're up in the air, but if you're not actually thinking about control, so you can get up, but can you actually control what you're trying to do? That's another thing to think about. And I think about it from a clinician standpoint. You give them an airplane that can get up, but can they actually steer it to the place where they want to go? And so, looking at the history of flight, the Wright brothers and that generation suffered this, challenge as well. How can we design a heavier than air aircraft when the principles of operation aren't understood? Well, some people are trying to hack it and actually put them at great risk and actually put them at their own lives at risk. And so the innovators in the field actually stopped. And first they built the wind tunnels, the lift balances and the like to understand the key principles of lift. And then they also took a mindset from the very beginning of not just understanding lift, but also what are the measurements and models that help us to control that system so that we actually can keep it going from point A to point B as we desire. So I think we can learn a lot. This is what I have a team that focuses on research tools. That's what we're trying to do within neuromodulation is as we think about how to improve things, a lesson learned for us is we need to start building scientific instrumentation to enable the next generation of products to be developed. It's not that we can a priori know what the answer is. So this comes down to strategy. What is the access point to the nervous system? There's of course a long history in the epilepsy space of bootstrapping off the EMU recordings during seizure localization and kind of taking advantage of that access to do measurements. Well, we see the same thing in the brain modulation space where a clinician has access in the OR to the brain and can do some quick measurements, plugging in, say, to a Tucker Davis or BlackRock system. But the issue is this is a very time-limited process, leads are externalized, and very artificial use cases. You're under the gun, subjects in a certain pharmacological state. They certainly can't get up and walk around and do regular activities of daily living. So what we wanted to do was take some of the best instrumentation that we could find in the operating room, in the spirit of BioCast, shrink that down with integrated circuit technology, embedded in the device. And then the goal is to get the data anytime, have it fully implanted, and that allows us to have activities of daily living that are more representative of the final use case. Now the key is, let's be honest, you've read some of my team's papers. The performance of an integrated circuit amplifier that's going in a device isn't anything like what you get from a Tucker Davis or a Grass amp. And so I think about this from the classical disruption curve, where the performance characteristic of the instrument is actually lower than what the clinicians are used to today in the OR. But what we're providing is new access points that allows them to gather data, and hopefully over time, like most disruptive technologies, we can make use of that lower grade instrumentation but get better overall system and product performance in the future. That's at least the intent. So this is part of our exploration of building tools for the BRAIN initiative, and I'm going to walk you through one specific example where we think about modifying the brain-machine interface technology that we hear a lot for prosthetics and how we're trying to apply that within the brain modulation space. And of course, there's a lot of instrumentation that goes about with it in the block diagram. I'm happy to take more questions on this at the Q&A. So our architecture, and this is actually a lessons learned, and someone was asking about the regulatory process. We were very intentional about this, about building a platform off of an existing neurostimulator, which basically firewalled it from the new scientific toolkit. And so that way, this measurement could be woken up, a clinical researcher could then take their data, and then we'd shut it down. And we really focused on minimizing the incremental risk of the baseline therapy. And so when we think about this as a payload, we had 2 laws that we had to abide by, design laws. One, do not compromise the core therapy, the core clinical pathway that you're leveraging to do your research. And then the second one was to ensure the instrumentation had the capabilities to justify the research itself. And so those were our two design criteria. But what we like, and you hear a lot of it, the challenge is, once you have this toolkit, you can actually scale it and put it in a device. And now we have chronic access to the nervous system. Give that capability to our research partners. So here's an example shared by Helen Bronte Stewart, who has an investigational device exemption for the system. These are some of the lessons learned. This is a subject who's come back. This is the telemetry head. And one of the mysteries is, in their different states, what's going on within the brain as they follow up. So say you have them go through activities of daily living, different motion tasks, which you can capture on the accelerometer. Now we could stream out that data in real time, looking at the frequency content of the LFPs, and correlate measurements. So for the researchers, one of their big needs is to synchronize internal and external instruments as closely as possible. Something we don't actually think a lot about, at least I didn't before we got into this, is one of the lessons, how do you synchronize, say, 5 different instruments and get them time locked, especially when you have an implanted system as well as external devices. But this is the first window into what's going on with signals on the order of a microvolt. This came up yesterday in Nitish's talk. These basal ganglia circuit signals, a microvolt signal is generous. Many of the signals that we're looking for are actually hundreds of nanovolts RMS, and so this can make it a challenging problem. Now what researchers are also interested in is then probing. So beyond just passive measurements, they want to actually actively probe the networks. So they want to use things such as a stimulation. So in this case, at about just after 5 seconds, they turned on 3 volts of therapeutic stimulation, and now they can go back and regather their information. And so here they can look at what happens to the lower frequency band, like that microvolt beta band, as well as look at other bands like gamma peaks. One of the things we had to add was a test tone, because of course you have to make sure when you have a 3-volt stimulation signal that you aren't actually just measuring artifact and saturating your preamplifiers. So there's also a lot of instrumentation kind of risk management you have to do in order to ensure the fidelity of the data is there. But with this capability, it leads to some exciting observations where looking at spectrograms and doing titration curves, just like we think about our circuits in the semiconductor realm, we can start to look at transfer functions within different disease states and within neural circuits. And that's what the researchers are starting to apply. So in this case, looking at the STN circuit, the team has published some of their observations on the different phenotypes that they see within Parkinson's, both within a specific patient and then across patient populations. But all with the challenge of building up reasonable instrumentation. The other thing that's interesting when we think about whether movement disorders is dynamic is, of course, subjects take medication. And so another team, the UCSF team, they have a investigational device exemption. And one of the things that's interesting in their subjects is they come in off their medication state, they'll turn on the loop recorder and basically measure neural dynamics that are occurring as the levodopa kicks in. As their artificial dopamine takes effect, they can look at the clinical assessment. So the clinicians can actually be scoring and keeping a record of the state of the subject, while at the same time they go back and then look at the diary and say what was going on within the neural network. This is an interesting discovery that the UCSF team has been sharing just recently at SFN, where they came in and said, hey, there's this very high Q gamma band that arises that's very different than what they've seen elsewhere, and it's correlated with the dyskinesia state. And so when we think about movement disorders not being dynamic, on the contrary, this is a case where they're exploring now building a closed loop system where they can measure the onset of this peak and then actually turn the stimulator down to avoid a dyskinetic state within a Parkinson's subject. And that's one of the other things, the takeaways from the Wright brothers is always having this idea of learning cycles. Because they started out, this is one of their first prototypes of a glider. And what they discovered is using what was known at that time, this was sort of the lift to drag ratio. And they said, something's not right. Actually, what's published isn't quite right. So they generated their own data and then refined their design, and this was the next generation of systems. And so I think we have to take that mindset as well. Andy reinforced that. This is not a linear process where on the first design, you're going to probably have an optimized solution. You need to actually build into your development process a series of learning cycles and tests and come at that from first principles. The other thing I want to put here just is a little awareness. Be careful in engineering when you go outside. This is an IEEE conference, so I can say control. When you go out into a clinical space, be very intentional with your language. Because imagine how one of our patients feels when you say, I'm trying to control your circuits, your brain circuits. I mean, I say this with the utmost seriousness. We really have to be careful in the translation space. One of my lessons learned, and I actually learned it at Berkeley from a gentleman sitting in the audience who got really upset with me about using the idea of programming the device and programming the parameters. So that's why I want to say, as we move into this spirit, learning cycles, it's all about building a more optimal, robust therapy. Let's not think about it like controlling the patient. So as part of this need for learning cycles, we've embraced the idea of that computer in the loop, where we could stream out signals and building research tools where we stream out data from the implant. We can then gather data from other externals, run a algorithm that does classifier and controls, updates, and then telemetry that back in real time to the stimulator output. It's this kind of flexibility, I think, which will help to rapidly assess the quality of different algorithms and their capabilities. And these systems have actually been applied in some pilot work. In this case, this is the Oxford team under Peter Brown, where you can build a very simple control loop. So this is a Parkinson's subject. We saw that low frequency beta band and how it responds to stimulation that's clinically relevant. They basically built a thermostat. No more complicated than a home thermostat, where the beta band is essentially like a temperature. When the beta goes too high, they turn on the stimulator. When it goes below, they turn it off. And what's interesting is still very much in the early stage pilot investigation, kind of arena, but they're seeing both a significant decrease in battery longevity, which a lot of us expected, but they're also seeing a reduction in some of the side effects. And so this gets back to that, be careful when we say it's good enough today. There actually might be a lot of potential upside in what we do. A lot of the stimulation parameters that we give today are not physiological by any stretch. These fixed patterns at 120 hertz. So let's keep an open mindset of how we might optimize things in the future. So this is kind of my wrap-up slide. Is we actually have had a very productive, I would say, public-private government partnership with the FDA and the academic community, where we're gathering up a large database of different neural targets and the signatures that come about with them, and starting to publish this database and making it available to the broader academic community to understand what are the similarities and differences of the signatures seen within these neural circuits. And this is just a snapshot of kind of where things are at today. Once again, all based on investigational device exemptions, but with a strong partnership with the FDA and academics. So that's just one example of the tool concepts that I think are important to help drive this field forward. And many of you are aware the NIH just launched a public-private partnership initiative, and we're looking forward, along with our other industrial colleagues, of helping to seed this initiative and really push the field forward by getting some of the translational science established. And the last thing I just want to close on is being realistic and optimistic. So a lot of this is saying, we don't know where to go, but we can't let that turn into cynicism or pessimism. I always like this quote from the New York Times, 1903. It's right after Langley crashed his airplane into the Potomac. And as you can see, And it might be assumed a flying machine might be evolved by the combined and continuous efforts of mathematicians and mechanicians from 1 million to 10 million years. So in 1 million to 10 million years, we'll be able to fly. Provided, of course, we meanwhile eliminate such drawbacks and embarrassments as the existing relation between weight and strength in inorganic materials. And 2 months and 8 days later, the Wright brothers took off. So I want to step back. I really think if we go back and build instrumentation, deploy it into the field, and just work collectively as an ecosystem to learn, we can be disruptive like the Wright brothers and hopefully move the field forward. So thanks.